So good morning, everyone. It's middle of August, so always it's a bit of a challenge to have a full house, but I'm glad that all of you are here to listen to Professor Dongsheng Liu, who came all the way from Tsinghua University in China. I just got back from China this Sunday. It's a phenomenal things are happening, and the pace as well is just phenomenal, both in science, engineering, as well as infrastructure. Actually, in Canada, we don't have that level of infrastructure, unfortunately. So, in terms of introduction, Professor Liu went from to do his bachelor's science degree in the University of Science and Technology in China. Then he worked in the Chinese Academy of Science, where he obtained his master's degree, followed by PhD in Hong Kong Polytechnic University under the supervision of Professor Albert S. Chan. In 2003, he went to Cambridge. I guess he worked with Mark Wellens' group as well, who is in our International Scientific Advisory Board at WIN. In 2005, he joined the National Center for Nanoscience and Technology in China as a principal investigator, and now moved to the Department of Chemistry at the Tsinghua University as a full professor. He was awarded a number of key academic awards, including the CCS RSC Young Chemist Award in 2008, the CCS BASF Young Innovation Prize in 2014. He was also the Fellow of the Royal Society of Chemistry in 2011 and became the Changjian Chair Professor in 2015. His research focused on DNA molecule machines and DNA-based smart materials. We're so happy to have you here and look forward to your talking.
Thank you, Sushanta. And it's my great pleasure to be in the Nano Center, the wing. And I appreciate very much for Joan's invitation. And it's my first time in Canada. But actually, I have already have a lot of connection with here because Professor Chen Wang is our former director in the National Center for Nanoscience and Technology. He's on the advisory board. And Professor Mark Wellens, who I work with, he paid me in Cambridge. And also I work with Shankar Balasubramanian and in chemistry. And so on the DNA I motif structures. That time I work on the molecular motors, and so which is a big important part in the Nano Center. And now is the molecular motors, the small molecular motors was awarded a Nobel Prize. But the big molecular, I mean DNA molecular motors is not included in. It's a pity. And I think Shankar maybe should be titled for that kind of part of the Nobel Prize. But it's in the future. So today I will talk about our recent researches. It's not on the molecular motors. Because years before, and when we talk about the self-assembly, because it's a very important method and we do to prepare the nanomaterials for the drug delivery and for the functional materials and also for the molecular devices. So how can we do the self-assembly? Normally we have a molecule, and when we talk about the self-assembly, and there's two methods. One is about you can just have amphiphilic molecules. It has two parts. One part is hydrophobic, one part is hydrophilic. So when you put them in a special solvent, they are uniform solutions. And then you change the conditions, you change some of the solvents, you change the temperature, you change the ion, you change some pH values. And then part of them become insoluble, and one part is become insoluble, and then they will aggregate based on this kind of insoluble parts. And so they pile it up and form the micelles, vesicles, tube-like structures, or nanosheets. Many kind of morphologies we can get. And so actually we said that we can control the self-assembly because they at the position we want, at the morphology we want. Because we can change this part. We can change the morphology, the structure of the molecule. Because as a chemist, that's our technology, and to change the structure of the different molecules, and then we can get different things as we want. And people think as we can control the self-assembly. So that's a very important topic called controllable self-assembly. So, but how can we say it's controllable? And normally we can just change things and we got this kind of things different. So, but I have a very challenging idea. That is, can we use the same molecule under the same condition, but got the different size and shape? But they're all very stable, and it's designable things. So sounds like it's the very, very difficult things. Because as a chemist, normally we use the same thing, and on the same condition, you got exactly the same final products. But here, we ask the different products, but on a controllable way. If it is not controllable, it's no use. But is that really exist such a system? The answer is yes, because in the nature, actually, the cell is a self-assembly system. Actually, when you look at the cell, the most important part is the cell membrane to us for the self-assembly, because it's lipid bilayer. Lipid is a small molecule, so they can form the bilayer. And normally, when we have a lipid bilayer, when you can get a bubble. The bubble is very, very fragile. When you touch it, it's blown up. And so it's formed the lipid bilayer. It's the same material, but same different shape and size. But they all have a different properties. Why? Because the cell is very, very stable. When you touch the cell, because our skin, our muscle, our whole body is made by the cells. But the cell is so stable, and you cannot just punched, blown up. And when you just touch something, it's quite stable. But this kind of bubble is what we do in our lab. It's very, very fragile. So why? And when you do use this kind of the vesicles and for the drug delivery, the most important problem is the stability. How can you just make it stable like the cell? So that's a question. And also this kind of thing, another question is the cell was composed by the same lipid materials, but their shapes, they can change freely. But they're all stable. So why? So when we learn and this kind of in high school textbook, and we were told there's a lipid membrane intercalated with a lot of membrane proteins. They are free floating in the membrane. So that's not true, actually. And actually, last century, in 1992, there's a science paper called this kind of skeleton in the cell. We all know that's a kind of skeleton that made by proteins as exists in the cell, inside the cell. And so the membrane proteins, they have a very strong interaction with them by a small protein interlocking to each other. So the membrane protein is not free-floating just in the lipid membrane. So how it works? And I think the mechanism is totally reversed. It's totally different. That's like that. We have a skeleton, and the pink color means the membrane protein, mimic the function of that. Actually, you can see the cell structure is like that. They have a skeleton, and we call it the frame. And then we just fix the points of the membrane proteins. And actually, these points decide the shape and size of the cell. Just how they can just make this kind of the shape determined like this. Just in between of different membrane proteins, the lipid was filled in. Actually, they are not ball-like structures. Actually, when you just fill in the gaps, actually 3 points, they can decide a small pieces of the flat surface. So that's a triangle. So actually, the cell, when you just look in detail at a nanometer size, they should look like a lot of, maybe thousands or millions of this kind of small triangles, the flat surface. So they make like this kind of polyhedron structures. The morphology is not like the surface, not a smooth surface, but they have some curve. So they have many, many curves. So when this kind of skeleton, the frame, they change the shapes. Actually, this kind of the angles in between of this kind of small flat surface, that will change. So it's very, very easy in the dynamics. So all this kind of structures, they're all stable under the physiological conditions. So that's the reason. The argument I have made, sounds reasonable. Maybe you agree with me. But a lot of scientists, especially in chemistry, they think if you are a chemist, you just only just make a kind of proposal. It's like some physicists that do such things. But the chemist always, you should just repeat this kind of process use an artificial system. Because chemistry, that means the chem is try. And you should try with the homemade materials. So I was asked to do so. And because this argument cannot just answer the question. So I made an artificial system. It's much simpler compared to the cell. But it has the very basic elements of the cell. You can see here, I use a gold nanoparticle to mimic the function of the cell core. And then I use the DNA double helix because it's quite rigid, and to mimic the function of the skeleton. So we call this a frame. And with the frame, and how can we use the protein that's in the membrane? So it's very difficult to purify. I asked the biologists to say, can you just make a protein, membrane protein, just to have a single-strand DNA modified on that? They say, it's possible, technically possible, but we won't just make it for you, because if we can just make that, we can publish several Nature papers. So why we do it for you? So as a chemist, we just have another alternative choice. Says we can do this kind of, we call the leading hydrophobic groups, because that will lead the self-assembly of other amphiphilic molecules. So it should be just mimic the function of membrane proteins. Think about the membrane proteins. It's big enough, it's hydrophobic at the side. So they are quite a stable structure, and when they on cytological conditions. So we try to synthesize some big molecules which is hydrophobic and attached to the DNA. So it's much easier. And then we can just guide the other molecules, just fill in the gaps and form the vesicles we want. So the chemist. Chemistry and always is much easier because we have this kind of idea. Say the size is big enough and it's hydrophobic. The benzene ring is hydrophobic always. And then we make a dendrimer. The dendrimer is huge molecules, but has a monodisperse molecular weight. So their morphology, their molecular weight, and their size and shape, they're all determined. So we design this kind of dendrimers, and then use this kind of chemistry. We can use a solid phase support synthesis and to make this kind of molecule together with the DNA, and they attach chemically cross-linked, linked to this kind of DNA sequences. And this kind of things is very interesting, and because we have considered the size and the hydrophobicity of the molecule, but unfortunately the CMC we cannot calculate. So all the CMC was measured. So what's the CMC? This is a critical concentration that can form a vesicle or this kind of mesols. So here, they don't form any kind of vesicles. They don't form the mesols, and here they form this kind of fibrous structures. So you cannot anticipate a molecule what they can form, because actually we call it controllable, because the destination is fibrous structures. We say it's controllable, but actually we want them to be a vesicle, but we cannot control that. So they form this kind of fibrous structures is a disaster. But we can write a paper say we can just control them and to form a fibrous structure. Yeah, that's a trick we always play, because when you say something, but actually we just shoot a bullet and we hit somewhere and we just circle this side, say, okay, we have this kind of, we got a target, 100%. But normally we want to hit the other target. But here is it. But we got something published, but it's not what do we want. So because we wanted a soluble molecule, and then we can just put them onto the frame. So we change. So as a professor, it's very, very easy, because I draw a new picture. Say we add some OEGs. OEGs is the water soluble. We add all 8 OEGs outside. So it will balance the hydrophobicity. And now you got is the molecule is soluble. But it's very hard to make. It's not my business. It's my student's business. So he spent 6 months to do that. But, the chem is tried always. Because we try to make this, we got this molecule, but the property is not that good because it's very difficult to be purified from the solution. You know you got this kind of material, but it's stick onto the column. So we have changed to the patch. I think it's driven, you run a lot of patch always. So, but when you just run patch for the material in the milligram and 2 gram size, so is the labor costing work. Actually, it's only labor costing. It costs nearly more than 3 months and for him and to just purify several hundred milligrams and from the solution. And so it's a hard work, but it's not that painful and only just for the labor. But finally, we got this kind of molecule. It's very stable in solution. And you can get a solution, is a uniform solution. And then we can put them together. I will not talk about how to prepare this part because Yuan is the expert to prepare this. And it's very easy, and we have 2 different DNA sequences on a particle. Normally, we can use them for sensing. But here, I use them as a frame. Here, you have this kind of, the longer one can hybridize with this kind of molecule, the DNA sequences together with a dendrimer, and then you should form such structures. This structure, because both of them are soluble, and we think it should be very, very soluble. And we can use the light scattering to measure the diameter of these 2 and compare them to see if it's finished or is the process going well. But unfortunately, we cannot. And here is what we design and what we want. So this time, we only got this, like they pivot up to each other. And so as they aggregate, they form a huge solid phase, and then they precipitate from the solution. So what happens? So after several months, we think about the reason, and we discuss with some experts, and we have no reasons, but the only explanation is about this structure. It's not a very simple frame. It's a superparticle. Actually, it's the particle formed by this kind of supermolecular self-assembly. And here, you see the gonad particle, they're quite small. Actually, the DNA double strand, they're exactly the same length. So all this kind of hydrophobic groups, they are accumulated on the very thin layer of the core outside the gonad particle. Actually, if you just only calculate the concentration in this kind of surface core, it's a very thin layer. And their concentration could reach the molar level. The molar level is much higher than the SMC again. So here, the superparticle, they're so hydrophobic, and so they interact to each other. So that's good, because we say this kind of aggregation, and we make this kind of structure that have a very strong induce of the hydrophobicity. So then we can just put the other molecules inside this kind of skeleton. But how? And the simplest way is you add all this kind of molecule, because the same molecule, they will interact to each other very strong. But actually, it's very difficult to prepare, and so students is not there and to risk that kind of things. Because if they put everything inside, and it means another maybe 3 or 4 months labor and to do that again. So we try this kind of small molecules, like lipid, like SDS, all these kind of things. Doesn't work. So it's so disappointing. But finally, I persuaded him, said, OK, we have a lot of things. And we think it's waste, because we have published that paper, but it's nearly no use. So in the fridge, you can try that, because some higher grade students, they failed on this project, but they have made these molecules. Why not you use them just directly in the system? This time we success. And so it get back to the pink color. The pink color is so beautiful. When I look at that, I was so excited. And because here, that means Because this color you can measure with the UV says is exactly the same color and with the DNA modified gonad particle. That means it's monodisperse system. We can cite the publication of Jevons and say it has been demonstrated many, many times. So here, if they form this kind of monodispersed vesicles, that should be like this structure. We have a gonad particle in the middle, and then the DNA frame, and then we have a polymer some outside. So like this. Use a TEM because you cannot only guess. You need the direct evidence. And here, and we use a TEM and see this kind of in the middle is a gonad particle, and outside is the polymer. In the middle, in between, is some blank. So actually, the DNA cannot be seen by the TEM. Here, we got this, and we called the light scattering. Here you see the radius is about 11 nanometers of the gonad particle. But actually, the gonad particle is, the diameter is 30 nanometers. But on the light scattering, sometimes it's much bigger than you see on the TEM. But you can see the increase after you modify it with the DNA. You add another 3 nanometers. And then after you finish the whole process, there's about 22 nanometers. But the increase is accurate. It's 11 nanometers. So here we can say that we have this such a vesicle, and it's precisely controllable this following the process. But how can we say we can just control the size precisely? And it's not only prepare a vesicle, because it's exactly the same what we have done 1,000 times before, and we call the controllable self-assembly. But this time we want to say something different. Let's use the same molecular system under the same conditions. We got a different size, but they're all stable. How can we just change the size? The easiest way is we change the gonad particle size. Then we follow the same procedure. We got a different size. But that's not very persuadable. I would like to say here, we use exactly the same gonad particle modified with DNA, but here we add another 20 base pairs onto this kind of frame. And here, normally we use the 20 base pairs, but this time we use the 40 base pairs. And then the final products you can just compare with the light scattering. See here the distribution of this kind of particle size, distribution exactly the same. But the radius increased 7 nanometers. Think about the Washington's very famous paper. It's most cited DNA paper, the double helix. So each of these kind of bases to another basis, when they're in B format, the B helix, and they are 0.33 nanometers each basis. So 20 bases means about 7 nanometers. So here I show you that is use the same molecular system under the precisely control the same conditions, following the same procedure, and you got a different size. And so the size precise can controllable and at the sub-nanometer scale. So it's a very controllable way. But how about the shape? The shape is actually we want to change the shape also, but change the basis, and because change from the gonad particle to nanorods. Nanorods is very easy to be prepared, and then you can just modify it with DNA and following the same procedure, and then we got this kind of tuber-like structures. And so they change the shapes on the same condition. But we think it's good, but the referee does not. Referee thinks this kind of tube-like structure has been seen before, and because in the other self-assembly system, and sometimes we can see these tube-like structures. So it's not very surprised. I argue with him, what's the surprise? What do you want for surprise you? And actually, we got some idea, but it needs time. So we got published in 2014, and then we used another 2 years and tried just to persuade him and say it's a very, very unique method. So what you think about what strange kind of shape, and you can just image and to persuade the referee, and he never seen by his eye. or it never happened in nature. So there's something very, very strange. As the cuboid structures. So when you just blow a bubble, actually you think the bubble is ball-like structures. You never seen a cuboid or cubic, this kind of vesicles. So we think if you use our method, we can make such a vesicles in solution. So the start point is the DNA origami. The DNA is a kind of very powerful molecule. You can just fold them into a cuboid structures, and then you have a lot of toehold outside. And then you can just follow the same procedure and do the self-assembly with our molecules, and then we follow the same procedure form a cuboid vesicles in solution. This time, no one has seen that before. Yeah. So I tried to persuade him, but we cannot just wait so long. And in 2007, we published the results because you can see here, this is a starting material, and this DNA origami has 20, 40 nanometers in cuboid. And then, following the same procedure, you got this final products. This kind of vesicles, they're cuboid. They have a nearly vertical angle here. You see, this time we are very, very happy and to see the same review. Exactly the same review, because he told us that is, okay, this is a structure I have never seen. But the frame-guided assembly I have seen before. Yeah. So he told us, and oh, it's very, very powerful. And but really, this time I agree with you. This method is powerful enough, and to just prepare something unique I never seen before. Yeah. So actually, you needn't just you have seen that is we have a very painful pathway and to do this self-assembly, and always. And the PhD studies always be like that. And because you want to do something new, you need to just face to a lot of difficulties. And after you solve these difficulties, you find the easy way to do that. That's, I think, is destination. Sometimes it's, I think it's painful, but it's very happy. And so my students found that actually we needed to prepare that kind of dendrimer and cost 6 months. So actually, he can prepare this kind of polymer. The PPO is a commercially available polymer, and it's 2,000 in molecular weight. And then you use a same chemistry. You just attach them to the DNA sequences. Then you follow the same procedure. This time, it's not that difficult. You don't have a participation because under the 4 degree, the PPO is soluble. So actually, you do the self-assembly at 4 degree, and then you increase the temperature to 36, 37, and then they form this kind of vesicles in a very beautiful way. No participation, and you can see the final products is exactly the same. So this time it only takes about 2 days because the DNA PPO preparation takes 1 day, and the vesicles preparation takes another day. So we published another paper in a very easy way after the publication of that paper in several months. Because this kind of experiment, the whole experiment is finished in 2 months. So it's much easier. And actually, you can do it much easier than before. So this time is, even myself, I feel it's a little difficult and to tell the referee says, now we do the difficult things much easier. And how can you just persuade them to publish in a high impact factor journal, say, this time is much easier. It's not more difficult. Always we say it's a challenging before, but here it's not a challenge. It's just a downhill process. Here we use this kind of commercially available sequences and attach it with some cholesterol. Cholesterol is this in the membrane, cell membrane. And here we put them together with the SDS. They form this kind of vesicles in a very rapid way. And now is we say this kind of system, firstly, we work with a dendrimer, which is very, very difficult, painful to synthesize. And then we demonstrate it can do block polymers. And now it's much easier, and you can do this small molecule system. So it's a general method. Actually, it could happen everywhere. And could happen in nature, and could happen in laboratory. And you can use them to prepare some functional devices. And since we made is functional devices like this, and actually we try to use this kind of things as a drug delivery carrier. So you can just load the RNAs, the DNAs, the proteins via hybridization, and then you can do the self-assembly. And during the process, you can put into hydrophilic medicines just in the middle. And then you put the hydrophobic on the surface. So it is a multivalency drug carrier. But I would like to tell you that it's not a good nano carrier. It's a very good nano system, but they carry a lot of the different drugs, and they are very stable, no leaking. And so something like it's good. We can finish there. But when we just work with the company, they found a fatal problems. That is no release for a long time. So for the drug delivery, actually, we want them have all these kind of drugs released after they just reach the target. But here, they can exist in blood more than 1 month. So they have no release. So something like it's no use. They say it's perfect to load everything there, but it never just release, and so we cannot use that. So I was so disappointed and so surprised. Say, I have got this published, but I say it's a very good candidate for the drug delivery, but we don't think about how to release. And then we just rethink the system, and this part has not been published. And so we add this kind of thiol bond here, and use a redox method. You can have them just release. Also, oxidation is the same. And you can just break in this frame, and then all this kind of drugs could be released. And then this time, we want to persuade the company says, now you can use that. But now it's still a problem. That's the cost. They have so many concerns. And they want to make it easier. They want to make it cheaper. And they want it for free. So always. But we want something that it could be used by scientists, because the basic science normally, and we only just, I think it's just work with scientists. It's much easier than with a businessman. But I will learn to work with a businessman. Yeah. So here, we try just to work with a scientist. And because what's the future direction of this controllable self-assembly? And we think it is, we want to challenge ourselves, because we challenge ourselves with this kind of three-dimensional cuboid structures. At that time, my students have a very, very brave idea, say, why don't we just make two-dimensional structures in solution? Because normally the two-dimensional self-assembly happens at the interface, not in solution. But how can you make that? He argued with me, said is if you can use the cuboid DNA structures to make this kind of cuboid vesicles, so why don't we just use a three-dimensional DNA origami, which is very classic, and to prepare a nano sheet just above this kind of basis? I say, you can try. So why not? Because we already have the system, and he tried this. And actually, we have this kind of DNA origami as a two-dimensional sheet, and we plant 35 our molecules on the surface, because it's classic. It has a toehold like that, and has been used for by many scientists. Then we do the self-assembly. We got this two-dimensional self-assembly double layer here. Here is it. You can see from this kind of the bare DNA sheet, and then you got this kind of full-size the self-assembly bilayers. And this kind of bilayer, actually, you can cut them into half, because you get rid of the pre-plant, this kind of immobilized DNA, this kind of hydrophobic groups, hybridizations here. You get rid of half. And then you can just cut them into half. And also you can cut them in a different way, like a triangle. So something like you got this 35 person in the soccer field, and then they get the position there, and then you got maybe 10,000 people getting into there, so they got the different shapes. And you change the point, you change the shape of the organization of the people. something like that. And this time, we can study the relations between the surface concentration and the solution's concentration. The solution concentration is much easier, as high as you can, not over the CMC. Otherwise, they will aggregate themselves. So now you can have this kind of things as high as you can, and then the surface density. And this time, we feel so lucky. And because 35 is the lowest density we can have on the surface. Otherwise, you cannot get this kind of self-assembly. I think if first time we try with 20 or 21, so if it is failed, we will give up this direction. Because they just have a try. So we spend some time work with the 35, we got his success. Then the student is so excited, and so he can try everything in the following. Otherwise, he will give up or persuade me to give up because I say it's failed. How can we find the way and to get out? And so this time we are so lucky. And then we can just, because we have this kind of very flat two-dimension bilayers of this kind of, can mimic the function of the membrane protein, membrane. So what can we use? Actually, we put the proteins inside because the protein is hydrophobic. So actually, it's easy to integrate them into this kind of hydrophobic bilayers. But you can imagine it's a little different with the vesicles or cell we have. Because normally in the cell, all the vesicles we prepared, actually they have some curvature. So the direction of the membrane proteins, they are randomly dispersed because you cannot just determine which direction on a ball-like structures, and so they are three-dimensionally randomly dispersed. But here in our membrane, they only have one direction. That's vertical to the surface. So that's only one direction of the membrane proteins. So think about what can we use that. Because in the Cryo-EM, actually, and when you just put this kind of single protein in the cryo samples, actually you have a randomly distributed three-dimensional structures. Then something, you use this kind of, use a TEM to see them, and with this kind of like the lighter here, and when you see here is a bar, but here is a dot. So you just guess the direction, and then you use a software to pile them up. And then you take about maybe 100,000 this kind of pictures, then you pile them together to go to the final structures like this. So they call it the Cryo-EM method. It's been awarded Nobel Prize 2 years before. But actually, we have the method that you don't need to guess the direction of the proteins. Actually, we give you a reference, internal reference. Here we put we use this kind of two-dimensional bilayers here, and then we put the proteins inside. So here you know the direction of the DNA origami. You know the direction of the proteins because they are vertical to each other. So here, now you still got a similar like structure here. This is the protein, this is the DNA origami. And then you get rid of the DNA origami part, and then you know the direction. And if the software already know the direction, they needed to guess. Actually, it costs a lot of time and efforts. But here now you need only less than 2 days and to just make a high-resolution protein structures. Other than compared to the others is about 3 months. The software running on a supercomputer, and it takes about 3 months. But now it's use a normal computer, it only takes about 2 days. So it's more than 1,000 times and then faster than before. But now is if you can just use other system, and also you can just even make them just in several minutes or several hours. So much faster than before on the Cryo-EMs. So now we are just working on the more and more examples. So I just take back message about why we call it the frame-guided assembly. Because actually it's inspired by the cell skeleton. But when I just, I have a very complicated, very long names for this method. But when I publish it, I got a suggestion from a friend, said you should just make the technology, the name, as short as you can, because it's easier to be remembered. I say this compared to the traditional method we use for the self-assembly, and like this is Edinburgh Castle. And you can see a lot of this kind of plates says they are piled by stones. So it's a very traditional method. But for some modern technology, and it's initiated from China, and say, because we are short of the stones in the past time, we have so many population. So Chinese people, they use this kind of structure. So they use some bricks and some woods and to make a frame. Then you mount the windows around. So something like here, you use the steels to make a frame. And then you mount this kind of glasses or walls around, and you make a building. Actually, it's much easier. It not depends on the strength of the material you use the cover surface. But this kind of stabilized strength is from the frame. So it's quite a similar process. That is, we use proteins, DNAs, all this kind of nanomaterials to build a frame. Then we mount everything outside. It does not just obey the rule that is self-accumulation. That's only obey the stability and determined inside. So like, because this technology called the frame building technology, the frame building technology. So we named our process as the frame guided assembly. And we use the uncontinuous frame. It can induce most of this kind of amphiphilic molecules into different shape and different size. And so you can precisely control the size. Actually, you can just change the shape as you want. And it's most important thing is a general method. I would like to say that is the most important thing. And because it could apply to the biological system and also could apply to our synthetic molecular systems. And I give the talk. Actually, the work has been done by these guys. And this two students, they initiated this work, and they do a lot of synthesis. But they are not very successful on the self-assembly because they're in the period and they need to graduate. They got their PhD, they do their postdoc other place. And Dr. Dong, and he finished the first example, and it's very painful for him. But he's very successful recently. Now he's a professor in the Institute of Chemistry of CAS this year. And just after 3 years, he finished his PhD. He became a full professor. And Zhiyong is a very lucky guy, and who do the PPO. And Chao and Yang, they do this kind of two-dimensional self-assembly. And because Chao just got the idea, but he left after 1 year. And we have so many things I need to do. And so Yang just continue his work. And now is nearly finished. And he come back to Beijing, and we will work together on this kind of Cryo-EM methodology and together. Finally, I would like to thank the funding support. It's very, very important. And money is not everything, but money sometimes is very, very important. And without money, there's nothing. Thank you for your attention.
Thank you so much. We'll open up for questions and discussions and comments on the presentation. Yes.
Oh, thank you.
You often show the bilayers in your system, right? So I remember there is one example where you use cholesterol, so the DNA is connected to cholesterol, and then you use sodium diethyl sulfate, SDS.
Yeah.
Do you know that you have bilayers? Because okay, the cholesterol is attached onto the negatively charged DNA.
Yeah.
And so that means that if if you have a bilayer, that means that the surfactant, which has a negative charge, also is going to fade the DNA. Whereas the cholesterol, we could view it almost as a...
Oh, it's quite difficult to see the cholesterol. And actually, if you use the two-dimensional structures, actually you can see, because they can also just apply to this kind of system. And actually you can see the different modifications and the different area. And because you can use, the DNA is a little different with the anchor point you use here. So you can use 2 different types of DNA. And then use, I think here, you can use a golden particle modified with different DNA sequences to just assembly with them. You can see where it comes from. Is bilayer or is monolayer.
And you can see it with the SDS and the cholesterol?
Yeah, yes, quite a similar thing.
Because what is neat also is that for surfactants, you have this packing parameter that dictates what you are going to do, right?
Yeah.
So for SDS, they form micelles.
Yeah.
But here you managed to induce SDS to form a bilayer.
Yes.
Which is...
Also with the lipid, with the SDS, you can see the heights of them, and so the difference. And you can just put them onto the microsurface, and you can see this just a monolayer or is a bilayer. So, but the vesicles here, that's only just form the bilayer because inside is still is a hydrophilic, is some water solution inside. So there must be a double layer, and otherwise they are not very stable.
But what is interesting is that you take surfactants that people have studied and they have explained that they can only form micelles. Or they only, and here you take them.
Yeah. We force them into form.
To form something that is completely different.
Yes. So together with the cholesterol. Yes. Yeah.
Can I?
Yeah.
Another question I had for you. So let's say you have a bilayer now.
Yeah.
But compared to a micelle where each surfactant can move separately from each other, here you have those anchoring points.
Yeah.
So that means that the surfactants normally are not as mobile. It must be much more viscous.
Well, that's a very good question. Actually, I work with another professor.
Can you probe it? My question is, have you probed the mobility of the surfactants?
Yeah, I think the mobility of this kind of system, they are much slower than the mobility in a free vesicles. Yeah. But we need to just experimentally prove that. And so now I work with a professor in Changchun Institute of Chemistry and Applied Chemistry. And he works on this kind of lipid membrane and together with the cholesterol. And the cholesterol density, because they form some anchor point. This kind of, they call the Iceland of the cholesterol. So that will just influence the mobility of the lipid, just exchange rate. And also the mobility in the film and outside of film. So I think it's between the vesicles, the exchange is much, much slower. But inside, so because they have so many grid, I think it's because they have quite a strong interaction with the small molecule we used. So the mobility of the small molecule must be delayed, I think, a lot.
There are some fluorescence probes. There is one that we have used at DIPIME that can be used for the rigidity of microenvironments.
Yeah, that's a good idea. We haven't tried that yet, not yet. Yeah. But I only have. Normally, I take 2 students a year, but I don't have a postdoc. Yeah, so that's only the people because I have 3 directions. One is molecular motors. Another is DNA hydrogel. We also just work a lot on the DNA hydrogels. But this is only just 1/3 of our direction. So I can put only just 3 students on this project. But they have a lot of questions to solve. And so I'm very open to work with other people. And if they're expert in that field, we can provide the materials. And so we can work together. That's the most important way, I think, is to science. So if we learn from the very beginning, it's very painful for the students and also it's quite a time-consuming, I think, process. Yeah. Yeah. Thank you.
So very nice work. And I just I don't follow very well at the early beginning when you try to describe.
Sure.
Could you come to the slides? Yeah. So what you mean is that you have a gold nanoparticle.
No, good nanoparticle together with the DNA double helix.
Yeah, then you have a DNA, have a anchor to this.
Yes.
Touch the DNA, then you have a framework. Then you have another layer of your molecule assembled.
Yes.
Layer.
Yes.
That's what you mean. So then your second polymer, what
Yeah.
you mean is a
PPO.
is hydrophobic.
No, it's, at a low temperature is hydrophilic, but it will just thermal triggered the property change. And so when you just have high temperature, it's become hydrophobic.
Hydrophobic.
Yeah.
So your DNA is anchored to the gold nanoparticle.
Yeah, yes,
Then you have a PO hydrophobic.
PPO, yeah.
PPO.
Yeah.
Then you PPO assembled, then you have another layer of DNA. That's bilayer.
No, the PPO, actually the polymer is quite hard to say is a bilayer. It's a kind of the kind of mixture because they will pile up just in the polymer film. Because we call,
Which species is bilayer?
but the outside is also another layer of the DNA, as exactly the same with this DNA. Yeah. So
And what's the third one? SDS.
yeah, SDS. So the cholesterol was attached from here with the DNA,
Oh, cholesterol can also attach.
attached to the DNA. Yeah, the chemical link.
Then
You
the
use
SDS, what's the function of SDS?
the function of the SDS form the shell. So they follow the guide of the cholesterol was in the layer of the
I see. So
SDS.
So it's monolayer SDS.
So it's not actually SDS should be like this,
Is it monolayer?
the head to head, or tail to tail, tail to tail. They form a bilayer because otherwise they are not hydrophilic inside, because the inside is hollow, it's containing a lot of water. So the solution is still inside. So they need two sides hydrophilic. So must be bilayer at least. Yeah.
So it's quite interesting. It's kind of bilayer form that, as you said, is a very big achievement is designed by
Yeah.
this gold nanoparticle.
Yeah. Normally, they can form a missile. It's a kind of so all these kind of things they get together, but it's solid inside. It's solid inside. So outside is hydrophilic.
So what you do is just using water solution of a gold nanoparticle and the water solution of your another molecule mixed together.
Yes.
And all the ratio between these two components.
Oh, actually, this kind of small molecules is much excess amount we needed, and then we separated it from the solution.
That's small molecule. That's how
Yeah.
DNA from the.
So here you can see the structure like this. This is the SDS, and like this. This is the DNA is the green here. So they support like a string. They just
So if you get some ratio change, you
Yeah.
can connect all your nanoparticle.
yeah, sure. Actually, at the beginning, actually you got this nanoparticle just affiliate to each other, but it's quite hard to control this kind of directions. But now is also we want to control the directions actually, and Nicholas Kotov, and together with Professor Zhi Yongtang, and they do a lot of this kind of use the particle and use the different modifications that are hydrophilic, hydrophobic, because they have this kind of aggregate. They can form the linear structures, two-dimension structures, and assembly.
Looks like you do not need to have use DNA. You can extend this technique to something else, some without the DNA.
Yes, you're right. That's a point. Actually, but we are familiar with the DNA, so it's much easier for us to handle the structure and the frame. But actually, now some people suggest that we can use this kind of protein structures, and also with some polymer aggregates. You know, this kind of polymer as a core, and you can just do the same thing. So there's a lot of other researchers, but our researchers, because like Juwen, and we have worked in DNA field for 20 years?
Almost.
Yeah, almost. And so we are too old to learn new tricks. But our students now, they do different ways. And so I just applause for them and say, okay.
No, what I just discussed is because DNA you have lots of molecular pair interaction. So you tend to other molecules, there's no that strong pair interaction, only hydrophobic effect.
Oh.
That could be some kind
I mean, if you can use this kind of nanotechnology to make a frame, maybe by silicon or silicon dioxide and such things, and then if you use a chemical modification on the silicon, actually you can attach some hydrophobic groups. It's the same. It's not the DNA, but the frame must be rigid. If it is too soft, it cannot just do the work. Yeah. So the separation of this kind of DNA is good because there is a negative charge. But otherwise, you use the other things that don't have this kind of negative charge, they will aggregate themselves. So that's, so we are lucky because we work with DNA. Yeah, yeah. That's true. Yeah.
Have you ever dissolved the core, like the gold core? If you dissolve it, I guess.
Yeah, we have tried that. Yeah, it's this, because suggested by referee say after you remove the core, a good core, what happened? So we use the cyanide to get rid of the good core, but it will collapse. They form fibrous structures again, because they will just get back to themselves. Something like I think it's army, and when they just as a soldier they can just form the order, they can form different shapes. But after they just take off the uniform, everyone's behavior is just randomly, and so they form a new society. It's not like army anymore. So that's, but it's quite hard to get rid of this kind of core. Actually, using the cyanide is normal, but in the control release, when you're in the vivo system, in vivo system, it's quite hard to use the cyanide, and so they poison everything.
There's one more question.
Okay.
One last question. So let's say I have my gold nanoparticle. If I have just one strand with a cholesterol, just one strand.
Yeah.
So then I form a micelle around cholesterol. Let's say I have two strands. I'm going to have two micelles. I crowd my surface, and at some point they must be close enough that you're going to form the bilayer. What is the shortest distance, or what is the density of how much density do you need to have to induce a bilayer formation?
Yeah, that's a good question. Yeah.
And if we go further, so the more DNA you have, and the stiffer the bilayer is going to be, right? So
yeah. Yeah. So actually, I have mentioned here, so maybe it's too quick. Here, and actually we use this kind of method. Say, here the density on the surface.
Oh,
so here, this is 80 nanometers. This is 100 nanometers.
yes.
So in between there is normally 10 nanometers, the lowest density we have for this molecule, but they have different molecular different density, and because their ability and to induce other molecules is a little different. So
what is the limit? How
to the molecules, and I only just comment just on the molecules we used is the 10 nanometers.
nanometers, but if you make 20 nanometers.
Yes, they will just they cannot just form the bilayers. Yes, in between.
It has to be 10 nanometers.
Yeah, it has to be.
Minimum, yeah.
Yes, yes.
From your experience.
Yeah, but it's measured just one by one.
And if you do 5 nanometers?
Yeah, it's much better. Here, we demonstrate it. It's much better, much quick.
But then it's going to be more rigid too, because you have more anchoring points.
Ah, that's a good point. We haven't measured the rigidity of the kind of membrane compared with the different density. I think you're right. Yeah, I agree with you. It's right. But if somebody can just measure the rigidity, I know that Mark Wieland can do that. Yeah, yeah. But I haven't persuaded him to do so.
Well, we have very good discussions and comments. So thank you so much.
Thank you very much.